All right. Hi everyone. Welcome back. So from this point onwards, we are going to be talking about cells. What's inside the cell, what it does, how it does, what it does. Today, we are going to cover bits of cells, 3 out of 4 bits in particular. We're going to talk about the cytoplasm, about the different organelles, and we talk about the nucleus. Okay. The fourth part of the cell itself, called the plasma membrane or cell membrane, is what surrounds the cells. Okay, it's almost like a broad, a wall, I guess, around the actual animal cell. Okay. We're going to talk about that wall and what it does, other than protection, which you have guessed from the wall, in next session. Today we're going to talk about the cytoplasm, about the organelles, and about the nucleus. So let's have a look at this cell of ours. This is a schematic representation of a cell itself, and you can see that there's a border. There's the border outside, and this is what we call the plasma membrane. Okay, I sometimes call it cell membrane, plasma membrane, cell membrane. It's exactly the same. Okay, now within these borders you will find all these different bits and pieces. Okay, now I want you to think of them as almost puzzle pieces. Okay, all of these puzzle pieces will do slightly different things, therefore, as you know by now, they will look differently. Okay, so we're going to talk about what are these structures inside the cells, what we called organelle, and we're also going to touch on this sea of molecules and water that is floating these organelles. Okay, so inside the cell, you almost want to look at think of it as a balloon filled with water and different bits inside. Okay, with the balloon itself being the plasma membrane, the water inside being the cytoplasm, and the little bits floating around are the organelles. Okay, all right. So while I showed you this really great schematics of a cell that's really clean looking and very uneventful, a bunch of scientists a couple of years ago got together and took some insulin secreting cells, some beta cells that you're going to talk about in a couple of sessions time as well, and put them under the microscope, sliced them up, and then rendered a 3D version of the cells themselves. And what they found is chaos. So the cell, while we are talking about a cell in terms of a very ordered, bits and pieces inside in a very nice way, the inside of the cell is actually a mess, and it looks a lot more like this than like that. Okay. So why am I telling you this? Because I really want you to understand and remember that the way that I'm teaching you cell biology, the way that I'm representing the cells, is actually very reductionist. Okay, and that the cells themselves actually jam-packed with all these different organelles, making sure that they can function at appropriate level. Okay, but that of course poses a conundrum, because if cells are so messy inside, how the heck do they know what to do and how to do that? That's the question for another session. Let's talk about the bits first. So the first bit that we're going to start off with is the cytoplasm, that water inside the balloon. The cytoplasm itself is the cellular content between the plasma membrane and the nucleus. Okay, so everything floaty-like, watery-like, that you will find between the plasma membrane, so the outside of the cell, and the nucleus is called cytoplasm. okay, or cellular milieu. The cytoplasm itself is composed of two things. One is the cytosol, which is actually the watery part, and the other are the organelles. Okay, so the cytoplasm itself contains the cytosol and the organelles. The cytosol is something we called intracellular fluid. What does that mean? Intra, inside, cellular, inside cell, fluid, meaning watery fluid. Okay. This fluid is not composed only of water, but it does contain different components, both inorganic and organic. So we're talking proteins, carbohydrates, lipids, as well as the inorganic substances, including ions. Okay, and we'll talk about these ions and the levels of ions as we go through the semester as well. Okay, so keep that in mind, that they're actually around in this cytosol. Okay, the chemicals themselves are either in solution or in suspended form. What does that mean? That they bind to something else. Okay, functionally, you'd think something that really doesn't have a structure, which it really doesn't, right? It doesn't have this rigid sort of squarish or longitudinal or whatever sort of structure. It kind of fits within the structure. It still has a function, yeah? And the function of the cytosol is to perform the metabolic reactions, okay? So all the metabolic reactions that you can think of, and some that you will actually talk about, not just within the BIOH111, but also within BioB, actually happen within the cytosol. Okay. An example of that reaction, for example, is making ATP, which is your energy. Okay. The organelles themselves are cellular structures with specialized functions. What the heck does that mean? The cellular structures means that there are structures within the cell with specialized functions. So they perform some different organelles will perform different functions. When it comes to organelles, I really want you to think of them as little organs. Okay, and that's really what organelles means. Okay, organelles themselves are performing different functions the same way that different systems within you will perform different functions. So for example, the cell will have a digestive system to digest different things within the cell itself, the same way that you have the digestive system, right, to digest everything that you eat. Okay. So it's the same thing, the same sort of logic. Organelles being little organs that perform specialized functions within the cell. So let's kick them off with introducing something we called endoplasmic reticulum, or ER. Okay. Structure of the endoplasmic reticulum is the network of membranes that form really these flattened sort of sacs and tubules all the way through the cytoplasm. Okay. So what I want you to imagine is you have your cell with the plasma membrane, you have your nucleus, right? That's on one side of the cell, and then all around them is this network of membranes. Yeah. And why is it a network of membranes, and why do we need them all the way through the cytoplasm? Well, because they themselves are important for two things. One is synthesizing protein and lipids. Okay, to synthesize protein, you will need rough ER. Okay, it's rough because part of that ER contains another organelle on itself called the ribosome, and we'll talk about ribosome in the next slide. The lipid synthesis itself actually happens on something we call a smooth ER. And lipid synthesis is, you can imagine that, for example, membranes themselves are very much lipid based, right? So you need to make sure that you're replenishing constantly this lipid, okay? Because lipid in itself can be food, it can grow old, it can be replaced, and to be replaced, you need to synthesize some of it, okay? So rough ER has the protein synthesis because it has the ribosome sitting on it. Smooth ER only synthesizes lipids. Another couple of smaller functions of endoplasmic reticulum that are actually quite important. We don't cover in BIOH111 so much of the first one, which is the site of quality control for protein folding. What does that mean? It means that when the protein is made in a sort of chain-like structure, and at some point that chain needs to fold into a particular structure in order to perform its function. Now, ER is able to recognize whether or not that folding is correct, and if it isn't, it actually makes sure that that protein is actually digested with using a proteasome, which is something that we'll talk about in a minute as well. Now, the other point that I wanted to make here is that the ER is actually a site for storage of one particular ion called calcium. Calcium storage is very, very tightly regulated because it's also something we called a secondary messenger, and we'll talk about that in a minute as well. So calcium itself needs to be stored into the ER and can be released when needed. I want you to make a bit of a note somewhere in your notes here to come back to this point, because we're going to talk about calcium and ER when we start talking about how is it that the muscle contracts, okay? So ER structure, network of these beautiful membranes that are all through the cytoplasm, and their function is to synthesize either protein, if ribosomes are bound to it, or to synthesize lipids, in which case it's called a smooth ER. It's also a site for quality control in terms of protein folding, and also an important point to make is it's also a calcium storage site. What about these ribosomes? These ribosomes that are a bit weird. All right, so let's talk about, let's come back to the ER. So if we go back to our little schematic of the cell, you will see these sort of flattened membrane looking cells. Can you see these? These guys are studded. They have little spots on them. Can you see little spots? These spots are ribosomes. The smooth ER is over here. This is the one that doesn't have any spots. You can see that there's no spots on them, okay? It's because it itself does not contain ribosomes. Now, we know that the ribosomes themselves are able to produce proteins, but they themselves are a large protein complex. So if you look at the slide before that, you can see a ribosome, an example of ribosome like this, sitting on top of a membrane over here, okay? Ribosome makes the protein, and as it's being made, it is shuttled into the lumen or inside of the endoplasmic reticulum, okay? So this is what happens in your rough ER, okay? So ribosomes themselves, large protein complex, they're a site of protein synthesis, and we will come back to this protein synthesis in the next session. They themselves can be free. So if you look into our schematics of the cell, you will see little dots sort of spread out in the cytosol. Yeah, these dots are what we call free ribosomes, and they are able to synthesize proteins that are found inside the cell. You also have these membrane-bound ribosomes, which is the ones that I've just talked about. They basically sit on the ER membrane. So they're attached to the endoplasmic reticulum, and they synthesize proteins that are needed for plasma membrane and for exporting it out of the cell. I'd like you to have a bit of a think of how is it that those two things come together, and why is it that you are making proteins in the first place. We'll come back to that point in the later session. So let's keep going with our organelles. Once the protein is made, it is packaged into these little vesicles. We call them transport vesicles. And they're basically blobs of lipids around them and stuff inside. Okay? These proteins that are inside the membrane will, from the ER, so here's our little rough ER, okay, be packaged into the transport vesicle, and the first point that they will encounter is this 3 to 20 stack looking organelle called a Golgi complex. The Golgi complex structure is basically what I want you to imagine is these flattened sacs that are sitting on top of each other. Okay? There's at least 3 of them. There can be up to 20 depending on the cell type. They're referred to as cis, medial, and trans. You don't really need to know that level of detail, but I do want you to know the function of Golgi complex. I often say to my students that the Golgi complex itself is like a bus stop. Okay? Once the proteins come to the Golgi complex, 2 things really happen to them. One is something we call post-translational modification. It means that there are different components that can be added onto the cell itself. So for example, there might be a protein that has a sugar added on top of the protein itself. Okay? That happens in the Golgi complex. But what you have also done with adding of this sugar, for example, you can also add lipids, is you almost have given a little stamp on the protein itself. And that stamp determines where is it going to go, which bus will it take, which part of the cell is this protein going to go to. So for example, if you're glycosylated, i.e. you have a protein that has sugar on top of it, you're most likely to be put on a bus that takes you to the plasma membrane. That bus being a vesicle, yeah? Here's our transport vesicle or membrane vesicle, okay? So these buses are the ones that are taking the proteins inside or outside of them, yeah? That are taking these proteins to various places inside the cell, either to a different organelle, for example, or to a plasma membrane. Okay. Sometimes these vesicles themselves take the proteins back to the rough ER where they started from. Okay. So Golgi complex, a bus stop. Everything comes into the bus stop, and the bus, the passengers themselves need to decide which bus to take to where to go next. Okay. Now, in order for them to know where to go next, what you need is a ticket. Yeah. Golgi complex basically gives them the ticket. That ticket being addition of sugar, for example, or addition of lipids on top of the protein itself. Once they have the ticket, they board a particular bus, which in this case is a vesicle, and that bus or vesicle takes them to their destination. That destination being either plasma membrane or a different organelle, for example, lysosome, or even back to the rough ER. Okay, so Golgi complex is a bit of a bus stop. Okay. Now inside the cell you can also find these funny jelly-looking things, and these things are called mitochondria. Now mitochondria themselves are quite interesting sort of beasts, because it's an organelle itself has 2 membranes. It has this sort of outer mitochondrial membrane that's quite smooth, and you know, it's something that you kind of think of when you think of a membrane or a wall. Yeah, but then inside of that outer mitochondrial membrane, you will find these guys, which is the inner mitochondrial membrane, that basically forms these folds, which we call crista. Okay, crista here. Now, these crista are actually really, really important, because what they do when you do this membrane that's quite folded in and out in a very small space, which is what outer mitochondrial membrane is, what you're doing is you're within the mitochondria creating more space for particular ribosomes to sit on. Okay, more space, and these ribosomes also contain something we call enzymes. Now, these enzymes are really, really important, because what they do is they take your food and they create ATP out of it, which is your energy. Okay? So the membrane, the mitochondria itself, the structure of it, with the outer membrane kind of holding it all together, but with the inner membrane ability to have this crista in enzymes sort of sitting on this really large population of membrane that's kind of bunched together in this mitochondria, you're able to produce a lot of ATP in a very small space. Okay, ATP is extremely important. It's important for different metabolic reactions, and it's also important for making sure that, for example, your muscles contract. Okay, so on a bigger scale, contractions of muscle is definitely what the mitochondria are enabling you to do. We will come back to the mitochondria itself in the muscle session. What I do want you to have a picture in your head when it comes to mitochondria is a jelly-like structure that outside looks quite smooth, but when you cut into it, has these folds of inner membrane that's called crista. And the reason for that is that that creates a lot more space for these enzymes to sit on. And these enzymes are the ones that produce ATP, which is what the function of mitochondria is. Okay, so what if you're a cell that kind of wants to eat? You want to make sure that everything is in homeostasis, even at that level. And you want to make sure that you have different digestive parts that are able to cut up things in different ways. One of the digestive systems within the cell is an organelle called the proteasome. Now, proteasome itself is pretty large cytoplasmic protein complex. What does that mean? If you look at the pictures here, this is from top view of the proteasome. This is a different sort of side view, filled in side view of the proteasome. And this is the side view. So it's pretty massive sort of complex. And what happens with the protein, what this proteasome is able to do is cut up proteins that are not folded properly, okay? The protein itself that needs to be degraded is labeled by this molecule called ubiquitin. Now, this is like a death wish for a protein, right? This happens in the ER, and when the ER labels a protein with a ubiquitin, this ubiquitinated protein is then sent to the proteasome. And as you can see, this hole in the structure of the proteasome is where the degraded protein goes into, and out of it come these protein fragments or peptides. So the proteasome itself is able to take something on one end, break it up inside, and spit it out the other end in different little pieces. Now, we've encountered proteasome already in the ER section. So go back to that slide and see a little proteasome sitting on the side of the ER. So we had a ribosome at the top, the ER, and on the side there was a little proteasome. as a part of the quality control of the ER. So that's what proteasome does. It is able to only take the unfolded proteins, so the proteins that are not folded correctly, and they have this death wish sort of sticker on them. It's called ubiquitin, and it's able to cut it up into different peptides. Okay, why would it need to do that? The cell itself is very self-sufficient. Okay, when you are using energy to make this protein, but it's not really folding properly, and now you've cut it up into different points, what you can do is take these protein fragments and reuse them to make another protein that won't have to be degraded. Okay, so reusing things within the cell is extremely important. So the cell has evolved a way of making sure that through food or through something like this, there's constant sort of supply of basic ingredients such as protein peptides. What about lysosomes? Lysosomes themselves are little membrane enclosed vesicles. Now these lysosomes are pretty cool. I call them a wastebasket. And the lysosomes themselves contain within them enzymes that we called hydrolases. They're called acid hydrolases because lysosomes themselves inside have really low pH. pH is down to sort of 4.5. And it ensures that these acid hydrolases, these enzymes, only work in the lysosome because it has such a low pH. Why would that be important? When the function of a lysosome is a basket of digestion, the cell itself puts everything into the lysosome to be digested. And by everything, I mean proteins, fats, nucleic acids, so things like DNA, foreign DNA, or polysaccharides, so things like carbohydrates. Everything comes into this lysosome for digestion. Okay. This is an important point to make because if you had these hydrolases, if you had this lysosome that basically spills out, okay, it has this, it has a little moment and opens up, right? And these acid hydrolases leach into the cytosol. Because the cytosol pH is different, it's about 7.5, these acid hydrolases don't work in the cytosol. They only work in the lysosomes. Which means that if there is some sort of perturbation or something wrong with the lysosomes themselves, and the acid hydrolases somehow leak out, what happens is that these hydrolases can't work in the cytoplasm because it's different pH. So it's almost like a preventative block to make sure that your digestive enzymes, so enzymes that are able to digest things, if they spill out, they don't eat the cell, okay, from the inside. So lysosomes, little membrane vesicles that have low pH, have specific digestive enzymes inside that are able to digest literally everything that comes into them, from nucleic acids to lipids to protein, as well as the carbohydrates. The third sort of system for cell to be able to digest things is this little organelle called a peroxisome. Now, this peroxisome actually is not contained within every cell, and immune cells have a lot of peroxisomes, whereas, for example, neuron cells don't have a lot of it. So they're smaller than lysosomes, and they kind of perform the same function in a way. What happens inside the peroxisome is that if you have a protein, for example, that ends up in the peroxisome, your peroxisome is able to oxidize it. When it oxidizes it, it basically shuts it down. It won't be able to work anymore. Okay, so it's very it's similar to the lysosome, but it does this oxidative part first, so that the catalase, which is an enzyme that works within the peroxisome, is able to cut it up. Okay, so 3 different organelles that are important for some sort of digestion, right? Proteasome, lysosomes, and peroxisomes. Okay, you yourselves need skeleton to keep you upright, to allow you to walk, to provide support. It's the same with the cells. The cells themselves have cytoskeleton. Cyto meaning cells, skeleton, skeleton. There's 3 different types of skeleton: intermediate filaments, microfilaments, and microtubules. As you can see, they all have different structures. Not just structures, but also where they are in a cell. So let's go through the intermediate filaments first. You can see 3 different cells, and these are those columnar cells that, for example, line your gut. Okay, and you can see that columnar, these cells themselves have this brush border, or these extensions of the cytoplasm on top of them. This here is the nucleus. Okay, and you can see that these blue sort of filamenty structures are called intermediate filaments. Okay, now these you see that these intermediate filaments all come to a specific point on the plasma membrane. Okay, these points I would really like you to remember, because these are the points that we're going to come back to when we start talking about cell-to-cell junctions. Okay, so intermediate filaments, they are there for elasticity and tensile strength, which means that they make sure that the cell are able to move when they in a monolayer, so there's multiple cells attached to each other. They're able to be elastic, so there's nothing penetrating through the monolayer, but it's also able to provide that strength so you don't pull it apart easily. Okay. What about the microfilaments? Now these microfilaments are a little bit different, because they're composed of a protein called actin. Okay, actin itself is a protein that we will come back to in terms of muscle contraction. But the actin is also able to kind of bring together, so you have the different actin made that bring together, and they're almost like chains. Okay, they're chains of protein. And when they do that, they form these filaments. Now, the difference between the intermediate filaments and microfilaments, it's not just the protein that they're made of, but also where they are in the cell. So microfilaments provide this, structural support and ability to transport things, and they're only found on the outside of the cell. Okay, so there's none of it inside. These microfilaments are only outside of the cell. Okay, now while the intermediate filaments composed of keratin, an example of protein called keratin, microfilaments are composed of a protein called actin. The third type of cytoskeleton is called microtubules, and these are actually the biggest ones. Yeah, they have a large diameter, 25 nanometers, whereas these guys are sort of in tens nanometers of diameter, right? So these are about 2 to 3 times bigger than, or thicker than, these guys. Microtubules themselves are composed of proteins called tubulin. Okay, and what these guys do in a very nice way is that they start at one point in the cell. Okay, and this point excuse me, is called microtubule organizing center, or MTOC. And we will actually come back to that particular point later on in the session. Now, what happens in the microtubule organizing center is, what I want you to imagine is a point of sprouting. Okay, so the microtubules are able from this point to basically grow out and retract if they need to. Okay, so they're almost like sprouting out and retracting as needed. Now, this skeleton in particular is very important in cell division, and we will talk about the cell division in a couple of sessions time. But it's also important in that vesicle transport. Remember those vesicles from Golgi and from ER that are carrying your proteins? Well, those vesicles need the grounding. The buses basically need the road. Yeah. Now, this road is actually provided by the microtubules as well as by the microfilaments. These microtubules actually don't go all the way to the plasma membrane. What happens is that your vesicle that kind of is going down this road here, for example, once it hits the end of that road, jumps over to the microfilament network, okay? And it then goes to the plasma membrane. So what I want you to imagine when it comes to cytoskeleton is not just the skeletal sort of function of being supportive, which all of them are, but also being able to provide space for a lot of functions that occur in the cells. One of those functions being the movement of these vesicles around the cell, okay? So, 3 different types of cytoskeleton: intermediate filaments, microfilaments, and microtubules. They all have different structures, therefore, you guessed it, they all have different functions. Microtubules that we just talked about also have a specialized little function, and that is they form something we call centrosomes, okay? Centrosomes themselves, remember how I told you that microtubules are involved in cell division in previous slide? This is how, because microtubules themselves will make this particular structure called a centrosome that enables the microtubules to grow from, yeah, when it's time for cell to divide. So we will come back to this particular organelle. So I need you to make a little bit of a note of what a centrosome is. So when I talk about the cell division later on, you know precisely what I'm talking about because you know what a centrosome is, okay? Now, we already talked about this a little bit when we were talking about the cells that line your gut, okay? Now, we talked about these protrusions, I guess, that you can see on some cells. These protrusions are called cilia, okay? You actually have cilia that can be numerous on the plasma membrane. And what cilia are is basically the plasma membrane prodded out by the underlying microtubules. Okay, so you almost want to imagine the microtubules growing out and taking the microtubule plasma membrane with them. That means that your cilia are actually specialized structure that are composed by the protruding microtubules and covered by the plasma membrane. Okay. Now, cilia themselves are quite short. They're sort of hair-like structures, and their function is to move things along. Okay, you almost want to imagine them constantly moving, almost like you know, the little anemones in Finding Nemo, where they're constantly moving and move the fish inside the anemone along. Okay. It's the same principle here. You have the cilia that are constantly moving, and if you have something, a particle, for example, sitting on top of that cilia, by cilia moving, it will move it out of that particular, on top of that particular cell. Flagella is similar concept. It's still microtubules protruding out that are covered by the plasma membrane. But now we're talking about one particular structure. It's almost like a tail of the cell, and that tail you only find in one cell in the body, and that's on the sperm cell. So both of these are specialized, I guess, structures that are composed of not just the microtubules that are protruding out from the cell, but they're also the ones that are covered by the plasma membrane. Okay, so cilia and flagella. So in the last couple of minutes, let's spend our time talking about nucleus. And nucleus is one of those big organelles that are sitting inside the cell. And let's talk about what it has in it and out of it so that we can understand the next session a little bit better. So, as I said previously, most cells actually have a single nucleus. There are a couple of exceptions, and I've already mentioned one of them, and that is skeletal muscle fiber or muscle cell that has multiple nuclei. So there's more than one nucleus in one cell. That's what we call a multinucleated. And then you have red blood cells. Now, red blood cells don't actually have a nucleus at all. Red blood cells are one of those specialized cells that really all they do is they want to be small and full of hemoglobin so that it can drive your oxygen around the body. So they don't really need a nucleus per se. Okay, they're short-lived, so they don't need to replace anything. So most of the cells though have the nucleus, one nucleus, and the nucleus itself is almost like a ball. And it's a pretty big ball inside the cell, okay? It usually does take up, in some cells it takes up most of the cell, in some cells it takes up, let's say, a third of the cell, but it's still a pretty big ball for a cell to have. Why such a big ball? Inside the nucleus itself is something you will find something called genetic material, or DNA, or chromatin. All of those are synonyms, so they all mean the same thing. Genetic material is DNA, and DNA, when it's nice and sort of stringy looking, it's called chromatin. Okay. In the nucleus, you will also find something called nucleolus. That's a small nucleus, and the membrane around it is perforated, so it has a little holes in it, and those holes are called nuclear pores. So I want you to imagine this ball that has a lot of sort of stringy stuff inside and has holes on the actual outside. Okay, so it's perforated, it has these pores that go inside the nucleus. This is communication point between the nucleus and the cytosol, and we will come back to that particular nuclear pore when we talk about the function of the nucleus in the next session. Now, the nucleus itself contains something we called DNA, or deoxyribonucleic acid, DNA. Now, DNA itself actually is double helix. What does that mean? It has two backbones that are linked together by something we call base pairs. Okay, there's only 4 different base pairs that you will find in DNA, and those are adenine, or A, thymine, N or T, cytosine, or C, and guanine, or G. So A always pairs with T, and C always pairs with G. Okay, so what you have essentially is the backbone like this, with little base pairs that go almost like in a ladder like way, and the whole thing twisted so that it allows you to have a structure of a double helix, okay, which is the structure that I'm sure most of you have seen before. This is an interesting point that I would like to make. there's about 1.8 meters of DNA in each cell. So we're talking about one tiny little cell that you can't see with your naked eye. Inside it is this thing called a nucleus. And inside that nucleus is 1.8 meters of DNA. So this cell needs to really tightly control how this DNA is structured within this tiny little nucleus. Okay? DNA is there as genetic material. Okay? Genetic material is a genetic material because it contains something we call a gene. Okay? Gene is part of DNA, and it's basic unit of information. And information is basically encoding for what we call proteins. So the function of nucleus is this, taking this information and making something out of it. Okay, so it contains the information, but what it makes with it is what we're going to discuss in the next session. In your DNA, in every single cell that you have that has a nucleus, there's a total of about 25,000 genes. Okay, 1.8 meters of it, 25,000 separate genes. Now, if you think about this conundrum a little bit, right? The cell itself has 1.8 meters of DNA inside the nucleus. And that can be arranged in this sort of stringy way, okay? Now, that's okay if you're just kind of existing and you're making this, these proteins, and you're just, you know, in everyday homeostasis. However, it poses a problem when you need to divide into two. So what the cell has done is basically taken this 1.8 meters, of DNA that's messy, and shrunk it or condensed it into something called a chromosome. Okay? So non-dividing cells, you will see these stringy looking DNA. In dividing cells, so the cells that are just about to undergo division, the DNA itself will condense into the chromosomes. How does it do that? You've seen these chromosomes before, right? These chromosomes are these X-looking things, all right? So let's figure out how is it that the cell does that? Well, it does that by taking DNA and wrapping it around particular proteins called histones. So let's have a look at, we have two different DNA molecules. This is, I'd like you to imagine a ladder between. We're twisting it around, okay? Oops, sorry. Twisting it around. Now it's a double helix. Now imagine 1.8 meters of this in each of your cells. To condense it to chromosomes, what the DNA does is it wraps around a histone like this, another histone like this, and then they themselves wrap themselves around it. This is what's represented in this picture. So this is your double helix here. These are your histones. This double helix first wraps around a few histones. Then those histones themselves wrap themselves around each other, and then those big ones start wrapping themselves into the structure that we call a chromosome. Now each chromosome looks like an X. In the middle of the chromosome is something we call the centromere. Remember the centrosome? This is a centromere. You're going to need to know both of those terminologies because we're going to need them for when the cell divides. One part of the chromosome is called a chromatid. So you have two sister chromatids in one chromosome. One of these chromatids comes from mum, the other one comes from dad. Okay. So chromosome structure is extremely important. You need to know it so that we can discuss the division of the cells later on. So chromosome has a centrosome, which is the central point of the chromosomes, and it has 2 different chromatids. Now, if you take the cell that has condensed chromosomes in, and you put it under a slide, you will see something called a karyotype. Now, these karyotypes, you can see that there's 46 chromosomes arranged in 23 pairs, yeah, with 1 pair coming from mum, the other one coming from dad. Okay, so 46 chromosomes is what you have in each of your cells, except in your reproductive cells, okay? And we'll talk about that when we start talking about the cell division. So each somatic cell, which is every other cell other than reproductive cell in your body, will contain 46 chromosomes arranged in 23 pairs, okay? These guys here, these X and Y chromosomes, are the ones that determine the sex of the sex of you. Females or girls have 2 of these X's, whereas the boys or males have X and Y. Okay. So that's it. We're going to recap the animal cell and what it has inside in the next session. See you then.